So, good morning, students. Stay safe, stay home. So, today I am starting with a very important lecture of lecture number 1 on the taxonomy, evolutionary, and systematic bacteriology. The key points which we are going to discuss are important scientists related to classification of plant bacteria, and second is a grouping of bacteria. Now, if we see here, in this grouping of bacteria, as if you see there are list of scientists, different taxonomists who have grouped the bacteria in various groups. Now, Linnaeus in 1735 he grouped bacteriology in bacteria kingdom, or actually he made two kingdoms basically, and they were Animalia and Vegetabilia, and minerals was the third group, but it does not belong to the life sciences group. Haeckel, 1866, he gave us three kingdoms: Protista, Plantae, and Animalia. Keton, in 1937, gave two empires, Prokaryota and Eukaryota. Copeland, in 1956, gave the theory of four kingdoms, which were Monera, Protista, Plantae, and Animalia. Coming to another scientist, Whittaker, who in 1965 gave the concept of five kingdoms. What are those? Monera, Protista, Fungi, Plantae, and Animalia. Then Voisetel in 1977. He gave the concept of six kingdoms, which are Eubacteria, Archaeobacteria, Protista, Fungi, Plantae, and Animalia. Voisetel, he gave us the concept of three domains, Eubacteria, Archaeobacteria, and Eukarya. So these were the different taxonomists who gave us the grouping of bacteria in and the different kingdoms. Now, if you see, when we read bacteriology, we find one of very important name, that is David Hendrick Spurgeon from 1860 to 1937. He was an American bacteriologist who was recognized for his profile and varied researches in the areas of tuberculosis, food preservatives, phagocytosis, and anaphylaxis. He determined some very important characteristics to differentiate and identify the organism of class Schizomyces. From 1932 until his death 1937, he remained as Director of Biological Research at the National Drug Company in Philadelphia. In 1923, he and four other bacteriologists, they published the first edition. And what was that first edition named as? It was named as Burgess' Manual of Determinative Bacteriology. It's a very important, fundamentally very important reference book for identification and classification of bacteria. Now, the first three editions of the manual published by American Society of Microbiology. If we see the subsequent editions, 4th to 8th, they were published by Burgess Manual Trust, which were based at the University of Georgia in Athens, USA. The 9th edition of the manual is replaced by the new title, Burgess Manual of Systematic Bacteriology. The first edition of this new title was published by Williams and Wilkins, Baltimore, MD, in 4 volumes. In 1984, Volume 1, in 1986, Volume 2, and 1989, Volume 3 and Volume 4. So, this is a very important part where I wanted to explain you. Now, we'll come to the other part of the classification, how the bacteria has been classified. So you all know this, that you all know that DNA sequencing technology has become a very important component present in the present world. Now, this technology, it has led to a major change in the way that prokaryotes are classified. Okay, so sequence analysis of highly conserved regions of the bacterial genome, such as small subunit RNA gene, now is the most preferred method for bacterial classification. Now, the words which are in bold, I will explain you in the other lectures. The recent concept of bacterial classification is what? It is based on the hierarchical systems like domain, phylum, class, order, family, genus, and species. as you see here. Further, I will show you that how in the Burgess Manual of Systematic Bacterial, this is actually a glimpse of second edition, 5 volumes. Like if we have to classify Erwinia keratopora, how we will place it in the, how we will write it. So here domain is what? Bacteria. Phylum is what? Proteobacteria. Class is what? Gamma proteobacteria. Order is what? Enterobacteriales. Family is what? Enterobacteriaceae. Genus Erwinia and species genus Erwinia keratopora. So if you go to this, you can see here, this is the hierarchical system for bacteria explained. And this is a glimpse for the second edition, which is consisting of 5 volumes. Domain bacteria, phyla 27 were there, classes 41, orders 88, families 240, genera 1194, and species 647 were described. Now, what is actually Burgess Manual of Systematic Bacteriology? It is regarded as a reference book. You know very well, you will know about it. The first edition, which was published by Williams and Wilkins, Baltimore, MD, it was edited by whom? John G. Holt, and in 4 volumes, that is from 1884 to 89. Now the current edition, that is the second edition, it is published by Springer, New York, edited by Garrity, George M. Brenner, Don J. and Krieg, Noel R., which contains 5 volumes that encompasses 6466 species, 26 phyla, 41 classes, 88 orders, 240 families, and 1194 genera. The detailed contents of the first and the second edition are given in the table for easy reference purpose. Let me show you those tables. This is just a glimpse of how this is the cover page of those books, you know, like I was referring Burgess Manual of Determinative Bacteriology. I'm just showing the glimpse of the cover page. Now, what are the basic contents in the different editions of Burgess Manual of Systemic Bacteriology? Let us try to understand that. So, in first edition, year 1984, 1 volume was published, the Gram-negative of general, medical or industrial importance. And the editors were John G. Holt. Then 1986, 2 volumes were published. The Gram-positive and other actinomyces have been dealt. Editors same. 1989, 3 volumes. The Archaeobacteria, Cyanobacteria and remaining Gram-negative. Same editors. 1989, the Archaeobacteria, Cyanobacteria and remaining Gram-negative, same. Then 1989, the actinomyces, same editor, that is John G. Holt. Now we come to the second edition. It was published in 2001, and in the first volume, the Archaea and the deeply branching and phototrophic bacteria was published, and the editors were Garrity George, who was the Garrity George was the editor-in-chief. Boone David R. was the author. Kestenholz Richard W. author. So these were the 2 authors. Okay. Now when we come to the second edition and year 2005, where 41 classes, 88 orders, 245, 1194 genera were published. It consisted of 2 volumes. The Proteobacteria, Gram-negative, the single largest phylum, 72 families, which consisted of 72 phylum families and 425 genera and over 1875 species. Now this was the largest phylum, which consisted of 72 families. Okay. Now it consisted of 3 parts. Part A consisted of introductory assays. Part B consisted the gamma proteobacteria, and part C the alpha, beta and epsilon proteobacteria. And who were the editor-in-chief? Garrity George, Don J. Brenner, Noel R. Craig and James T. Staley were the authors. Now coming to 2009, there were three volumes. The Firmicutes, which consisted of gram-positive bacteria, and the editors were Paul D. Box, George Garrity, Jones, Noel R. Krieg, Wolfgang Ludwig, Fred A. Rennie, Karl Heinz Schläfer, and William B. and Witt. Again in 2009, there was another 4 volumes, and which consisted of the bacteroidetes, Planktomyces, Chlamydia, Spirochetes, Fibrobacterius, Fusobacteria, Acidobacteria, Verumicrobia, Dictyoglomi, and Gametimondidis. Who were the authors? Noel R. Krieg, James T. Stelly, Brian Hidboon, Bruce J. Pasteur, Naomi Ward, Wolfgang Ludwig, and William B. Whitman. Then in 2010, in the fifth volume, it consisted of Actinobacteria. The authors and the editors were different. Michael Goodfellow, Peter Kampfer, Hans-Jürgen Buss, Martha Trujillo, Kenichiro Suzuki, Wolfgang Ludwig, and William B. Whitman. So these, now at least now we can understand what is this Burgess Manual of Systemic Bacteriology has different contents in the different editions. Now coming to the another part of this lecture, we will come to another part, that is different phylums. Now out of 26 phyla, the phylum Proteobacteria, the phylum Firmicutes, and the phylum Actinobacteria, they are discussed in the following section. They will be discussed. And these three phyla, because they mainly contain what? Both beneficial or pathogenic bacterial genera to the plants. Now the commonly known bacterial genera are provided with boldface. Now phylum 14, what is it actually consisted of? Proteobacteria gram negative. Now if we come to the Proteobacteria, exactly what is actually the characteristic of the Proteobacteria? It is a major phylum of gram negative bacteria which include a wide variety of pathogenic genera and many others. They are free living, non-parasitic, and include many of the bacteria which are responsible for the nitrogen fixation. Carl Weiser, he established this group in 1987 and called it informally as a purple bacteria and their relatives because it has a great diversity of the forms, and it was named after Proteus, a Greek god who is capable of assuming many different shapes and is not named after the Proteobacteria genus Proteus. So now we are coming back to the same this thing, that how this phylum Proteobacteria has been divided into 5 classes. What are those? Alpha Proteobacteria, Beta Proteobacteria, Gamma Proteobacteria, Delta Proteobacteria, Epsilon Proteobacteria, which is based on the rRNA sequences. Now the important plant pathogenic as well as the beneficial bacteria which belong to different classes in the phylum bacteria are given in. Now I'll show you in the table 2. This is just a glimpse of some of the very important, bacteria like Azospirillum, then Pseudomonas and Xanthomonas, which belong to this particular group, that is Proteobacteria. Now coming to this important bacterial genera, which are the important bacterial genera in different classes of phylum Proteobacteria. What is Alphobacteria? It is having how many orders? So we can just have a look that the different orders under this if I see what is Alphobacteria, they can grow at very low levels of nutrients and have unusual morphology such as stalks and buds. Others include agriculturally important bacteria which are capable of inducing nitrogen fixation in symbiosis plants. Now coming back to our the table, here we see that the different orders. What are those orders? Number 1 order, that is Rhodospirillales. Now we see here that there are important 2 families, Rhodospirillacea and Acetobacteriaceae. What are the genera which are very important here? Rhodospirillum, Azospirillum, Rhodospira, Acetobacter, and Acidomonas. Coming to the next order, Rickettsiales. What are the families? Rickettsiaceae, Erlisiaceae, Holospiraceae, and the important genera are Rickettsia, Anaplasma, and Holoplasma. Now the third order, which is of importance for us, is the Rhodobacteriales, and belonging there is only one family, that is Rhodobacteriaceae, and it consists of Rhodobacter and Sulfobacter. Rhodobacter and Sulfobacter. Now Sphingomonidae, it is consisting of the family Sphingomonidaceae, consisting of Signomonas, Rhizomonas, Erythromonas, Erythrobacter. So kindly read and learn these names. These are very, very important names, and as you all know, students, classification is a very important subject, and remembering the genera is another very important aspect for a student. Actually, they may be, you can be asked any questions, so you have to be very careful while learning these genera. So which family do they belong, what is the order, what is the phylum, so you have to really understand all this. Okay, so another is Rhizobiaceae, which are the important genera: Rhizobium, Agrobacterium. Then Bartonella, Brucellaceae, Brucella, Phylobacteriaceae, Phylobacterium, Methylocystaceae, Methylocystis, and Beijeringiaceae, Beijeringia. Now coming another family, which is Bradyrhizobiaceae, Bradyrhizobium and Nitrobacter. Hyphomicrobium, Methylobacteriaceae, Methylobacterium, and Rhodomyces, Rhodobium. So this is what we have studied now. One class, that is alpha proteobacteria. Now coming to beta protobacteria. Now what is actually beta proteobacteria? It is a highly metabolically diverse and contain chemo, litho, autotroph, photoautotrophs, and generalist heterotrophs. So coming back to this original table, we come back here and we see in beta proteobacteria, Burkholdereales is consisting of Burkholdereales, okay, Burkholderiaceae, Ralstoneaceae, Oxalobacteriaceae, Alkaligenaceae, and Comamonadaceae. So what are the important genera? Burkholderia, Ralstonia, Oxalobacter, Alkaligenes, Comamonas, and Acidophorax. Now these are the important genera belonging to the order Burkholdereales. Now coming, which is having 1, 2, 3 important 5 families. Again we come back to the Hydrogenophiliales. It is consisting of Hydrogenophiliaceae, family Hydrogenophiliaceae, and the genera are Hydrogenophila and Thiopacillus. Coming to Methylophiales, consisting of Methylophilaceae, and what are the important genera? Methylophilus and Methylobacillus. There is another order, Neissereales, having Neisseriaceae and genus Neisseria. Nitrosomonadaceae, and what are the important genera? You have to really remember all of them, okay? So just don't escape all of these. Nitrosomonas, Nitrospora, and Spirilla. Which is the other family? Spiralaceae and Gallonella. Gallonellaceae, which is coming under this Gallonella, and Spirillum is coming under Spiralaceae. So then coming to Rhadocyclales, Cyclocyclaceae, and Rhadocyclus and Propionobacter. So then third is a very another important gamma proteobacter, which is another this thing order of this very important, and these are the very important, and they are the largest class in terms of species with validly published names. The type order is including Pseudomonas, which include the genus Pseudomonas nitrogen fixing Astrobacter, which is a well known bacteria, as you all know. So let us come back to our original table. Gamma proteobacter consisting of Chromatiales, Chromatiaceae, and Chromatium, Nitrosococcus, Thiococcus, Thiospirillum, then Ectothyrodospiraceae, Ectothyrodospira, and Nitrococcus, Xanthomonadaceae, consisting of Xanthomona, Pseudoxanthomonas, and Xylella, then Cardiobacteriales, Cardiobacteriaceae, Cardiobacterium, Thiotrichales, Thiotrichaceae, Thiothrix, Bigiota, Lycothrix, Macromonas, Thiobacterium, Thiospira. Then what is it? Rickettsiaceae, Pisci Rickettsiaceae, which is having a Pisci Rickettsia, and Francisellaceae, Francisella. Legionellales, Legionelaceae, Legionella. Coccillaceae, Coccilla, Rickettsia. Then Methylococcales, Methylococcaceae, which is considered of Methylococcus and Methylobacter. Then Oceanospirillales. And if we see here in Methylococcaceae, there are another two important genera, Methylomonas and Methylospira. Now coming to this order, Oceanospirales. Which are the important families? Oceanospiralaceae, consisting of Oceanospirillum, Halomonadaceae, Halomonas, Alcanivoraceae, Alcanivorax. Now coming to Pseudomonadaceae, Pseudomonas, Azotobacter, Rhizobacter, Xylophilus, then Moraxillaceae, Moraxilla. Alteromonadaceae, and which are the important genera? Alteromonas, Pheromonas, Marinobacterium, Marinobacter, Psychochromonas. Then coming to another order, Vibrionales. Family, Vibrionaceae. Genus, Vibrio. Aeromonadales, consisting of Aeromonadales, Daceae and Succini Vibrionaceae. What are the important genus? Aeromonas, and this family is consisting of Succini Vibrio. Then coming another order is Enterobacteriales. Family, Enterobacteriaceae. Which are the important genera? Escherichia, Brinaria, Enterobacter, Arvinia, Klebsiella and Pentoia. Pectobacterium, Salmonella, Serratia, Xenorhabdus and Yersinia. Then another order is Pastorales, Pastoralaceae. What are the important genera? Pastorella, Actinobacillus and Haemophilus. Now when we come to the another class, this thing is again when we are coming to this very important phylum, and when we are dealing with this thing, so we have already dealt how many? Already we have dealt 3. Okay, how many have we dealt in this phylum? 3 classes. Okay, now coming to the, we are now coming to the 4th one. So what is that 4th one? Delta Proteobacteria. What is it? Delta Proteobacteria, which includes bacteria that are predators on other bacteria and also important contributors to the anaerobic side of the sulfur cycle. Okay, so I'm coming back to the original table where it is clear what is, this is the class, what is Delta Proteobacteria, and order Desulfurales, Desulfuralaceae and Desulfurella. Desulfovibrionales, Desulfovibrionaceae, Desulfomicrobiaceae, Desulfohalobiaceae. And what are the very important genera here? Desulfovibrio, Desulfomicrobium, Desulfohalobium and Desulfomonas. Then coming to the order Desulfobacteriales, Desulfobacteriaceae, Now what are the important genera? See, you can remember them very nicely because somewhere the words, the names are somewhere interrelated. So when we can read here, it becomes easy to remember. So here it is very easy to remember. Desulfobacter, Desulfococcus, Desulfonema, Desulfobacterium. Here Desulfofuromonadales, Desulfumuranidaceae, Desulfuromonas, which is sulfur-reducing bacteria. Geobacteriaceae family consists of Geobacteria, which is a ferric iron-reducing bacteria. Then Pelobacteriaceae, Pelobacter, syntrophic. So, now coming to the Syntrophobacteriales. Syntrophobacteriaceae, Syntrophaceae, Syntrophobacter and Syntrophus. Then coming to another order, Delovibrionales. Family Delovibrionaceae. Genera is what? Delovibrio. Then another order, Myxococcales, which is having how many families? Myxococcaceae, Archanigiaceae, Cystobacteriaceae and Polyangiaceae. What are the genera? All related to the families. You can very well understand them and also learn them. You need to learn them by heart. So this is Myxococcus, Archangium, Cystobacter and Polyangium. Now another very important class is Epsilon Protobacteriaceae. Now here what is actually Epsilon Protobacteriaceae is important because it is It is often slender gram-negative rod that are helical or curved, and it is coming back to the coming back to our original table. We come to that Campylobacteriales. So this is the order of this particular class. And now if we see here, Campylobacteriaceae, which is having Campylobacter and Helicobacter. Now coming to the another family, Helicobacteriaceae. What is the genera? Vollinella. So we have really understood till today, we have understood till phylum. What is this? Alpha Proteobacteria. So we have come to the very important very important phylum. We have dealt with a very important phylum. What is that? Phylum Proteobacteria, which is having 5 classes. Okay, and now which has been dealt, the orders have been dealt, family and genera. So students, kindly read this and try to understand and remember them, because now I will be in another lecture, I will be dealing with the another phylum, and you all very well know what are the basic characteristics of this. You have understood actually what is the basics of the Proteobacteria. What are they are basically they are the gram negative, generally phylum of the gram negative. So they include a wide variety of pathogenic genera, which I have already dealt in that particular table. So thank you very much. Best wishes. Namaskar.